Morning, everybody. Today I'm going to be presenting a case on a 13-year-old boy who had T-cell ALL. He presented to the emergency room with acute onset right arm and leg weakness, for which a stroke code was called, and that is how I and the JAR working with me ended up seeing the patient. He was at home when he suddenly lost voluntary control of his right arm. He found his arm writhing and moving towards his head. He then developed numbness on both sides of his body from the neck down. When he got up to stand, he noticed that his right arm and leg were both weak and then ended up coming to the emergency room. When he got there, he ended up having right arm and leg weakness as well as ataxia in both of those limbs on physical exam. In terms of his relevant history, he had received a treatment of PEG asparaginase the day before, and about a week prior to that, he had received intrathecal methotrexate as part of his chemotherapy regimen. As part of his workup, he did receive an MRI, which ended up showing diffusion restriction in the bilateral centrum semiovale, as you can see here. When reviewing this pattern of imaging, it is consistent with cytotoxic edema and most consistent with a toxic leukoencephalopathy. And in this case, this was likely related to the methotrexate that he'd received a week prior. Neurotoxicity from methotrexate can manifest in a multitude of things such as stroke, seizures, a chemical meningitis, as well as an acute or a chronic leukoencephalopathy. Typically, you see white matter changes in the centrum semiovale or the corona radiata. And most commonly, neurotoxicity from methotrexate occurs with intrathecal administration, but can also occur with high-dose IV administration. Just to review methotrexate, kind of harkening back to basic biochemistry, methotrexate is a folate analog, and it acts by inhibiting dihydrofolate reductase, which is a key enzyme in the 1-carbon pathway for helping to create amino acids and the backbone of DNA. The mechanism of neurotoxicity is not fully elucidated, but several different biochemical pathways are likely implicated. Methotrexate is known to cause increased homocysteine levels, and this is thought to possibly cause a small vessel vasculopathy. Methotrexate also causes interference with various transmethylation reactions, which is thought to cause demyelination. And with methotrexate, you can also get increased adenosine levels, which can cause cytotoxic edema. Looking at different treatments in the literature, the one that's most commonly thought of is leucovorin or folinic acid, which is a tetrahydrofolic acid derivative that bypasses the inhibition of the enzyme. However, there are a couple of other treatments as well, including dextromethorphan. And the way that this works is that since methotrexate increases homocysteine levels, homocysteine metabolites are actually NMDA receptor agonists. So it's thought that some of the neurotoxicity might be mediated through the NMDA receptor. Dextromethorphan is a non-competitive NMDA antagonist, and that's how it's thought to help with methotrexate-related leukoencephalopathy. There's also some reports of aminophylline helping as well. And this works via adenosine. So methotrexate increases adenosine in the CSF, and aminophylline is actually an adenosine receptor antagonist. Now, going back to our patient, he actually made a full recovery the very next day and was discharged home without any deficits. And this is often the case with this particular process, that patients fully recover in about 1 to 7 days. However, some do develop chronic leukoencephalopathy with subsequent administrations and permanent neurologic deficits. With acute leukoencephalopathy, it's not likely to recur on subsequent treatments, and he did end up receiving subsequent intrathecal methotrexate as part of his regimen. Here are my references, and thank you very much.
Thank you, Tanziyah. So we're a little ahead of schedule. The food showed up late. We're going to take about a 3-minute break for people in the audience to come down and grab their tasty breakfasts, which are available for anyone at home should they come in. Why don't you set up while we're Come on in. I got to introduce you. Yeah. Who's going to sit here?
It looks like the hot seat.
Yeah, let me. Yeah, get some. I don't think we can play with her. She's a resident. I've been thinking about it.
Really?
Yeah, I think it's frost. Hide floating meeting controls. Okay. I know, Andy. We'll make her a fellow, and then it'll be fine. Well, let me know if we are not zooming. Okay. Put the CME in the code to Bax Judd. And you can go get yours first.
I'm just going to get a. All right.
All right, so we're about finished, what do they call it, hoovering up the food down here, and we'll get going again. Andy's over getting a cup of coffee, but I didn't realize, Andy pitched in at the last minute to give today's talk, and I didn't realize it was World Alzheimer's Day, so thank you again, Andy. So Andy Liu's a member of the memory, or we call it memory, we don't call it behavioral. He's a member of the memory group, and he's really thrived at Duke. He's taken on a variety of roles. He's a great doctor, and we're thrilled to have him here. He's currently assistant professor of neurology. Andy did his MD at Rosalind Franklin. Where's that?
Chicago.
And she didn't even go there.
She did not.
So why'd they name it after? I know she's an important person.
CEO is British.
There's a British person there? Okay. Yeah. And she was the only British person you could think of? Now, everyone must know Rosalind Franklin was. She was one of the people intimately involved in the discovery of the structure of DNA and took the X-ray crystallography image that was the secret to unlocking the structure. She was working either independent of or with Maurice Wilkins, who at Oxford, I believe, and Watson and Crick were at Cambridge. And her X-ray crystallography got Wilkins the Nobel Prize, but not her. She died young, I believe, of ovarian cancer, if I'm not wrong. Yeah, but a seminal figure. He then went to Yale, and then for some reason did a behavioral fellowship, which you don't really need because the patients come in and tell you, you know, my mom has a memory problem. So maybe that's what the fellowship was about. All right. And thanks for Andy. He's involved in a whole bunch of things. He's now the lumbar puncture expert, and he's also interested in tuberous sclerosis as a model of Alzheimer's disease. But he's also involved in a very interesting project which is now acronymized to MERIAD, Mechanisms in Epigenetics to Explain Racial Inequalities in Alzheimer's Disease. And Andy, I will turn it over to you. I don't want to hit stop share, but I do want to get out of this. All right, it's all yours.
Thanks, Rich.
All right.
All right. Good morning, everyone. So, as Rich said, I am an assistant professor in neurology and pathology, and today I'm going to talk about the Myriad study. The mic right here. Yeah. Thank you. So, all right. So, I think there's going to be questions at the end that people can put in the chat, or feel free to stop me at any time to talk about any questions that might come up. So, start off with the fun stuff, the disclosures. So I'm a consultant for the Darwin Research Group, but otherwise no conflicts of interest. So this is kind of my outline in regards to the talk today. I think because it's World Alzheimer's Disease Day, I'm going to talk a little more about Alzheimer's disease and kind of how it evolved and how we got to where we are today. A little bit of history, I think, is relevant today. So, But because it's relatively early in the year, I just want to define dementia, I think for everyone in the audience and in terms of clinical care, what does dementia actually mean? Because there's always a discrepancy between what is dementia, what is Alzheimer's disease, is it the same thing? And so anyone who's ever rotated with me in the clinic knows this table very well, because I show this every single time. And I'm always trying to determine someone's cognitive status from one end with normal cognition all the way to dementia. And so dementia is a clinical term. So it's not used in research. We use it at the bedside or the bedside saying, the bedside saying that brain function problems prevent someone from living independently. So this could be behaviors, this could be language, this could be memory. You know, is their memory so bad that they can't attend meetings and remember what's going on? Is the language so severe that they can't give a presentation anymore? That is actually dementia, and that fulfills impairment of their daily function right there. So that is the definition. Cognitive testing, you know, the SLUMS, the MMSE, the MoCA, all that is supportive of the diagnosis. I wouldn't hang my hat only on a score because these scores are skewed. The MoCA is skewed towards memory. And so people with dementia with Lewy bodies and frontal temporal dementias, they actually score normally on the cognitive testing. That doesn't mean they're normal. So just keep this in mind. Dementia is defined as this, and Alzheimer's disease is just the most common form of dementia. I'd like to move on to Alzheimer's disease now. So Alois Alzheimer's was the person that described these findings with the disease that now bears his name. Thank you. Perfect, thank you. So Alois Alzheimer's was the one that described this disease more than 100 years ago. Initially, he was a German psychiatrist and neuropathologist, and he followed Augusta Dieter for several years. She was this 51-year-old German patient who started having memory difficulties. And the interesting thing is that she was placed in the psychiatric hospital that Dr. Alzheimer's was working at. Her husband couldn't afford it. So he wanted her transferred to another hospital, but Dr. Alzheimer's intervened and thought that her presentation was too interesting, so he made a deal. He would cover the cost of her hospitalization if he could just look at her brain after she had passed. And that's exactly what he did in 1906. Probably not an IRB at that time, that's correct. So, but in 1906, that's when he described the extracellular plaques, the intracellular tangles that define this disease that now bears his name. And so with these neuropathological findings, along with a lot of genetic models, has led to the amyloid cascade hypothesis. And so a lot of our therapies and new drugs are really based on this amyloid cascade hypothesis with some controversy, but I'll tell you why that there's a lot of evidence for this amyloid hypothesis. So the amyloid first is cleaved, APP, amyloid precursor protein is cleaved by base 1, shown in these scissors here. It's again cleaved again by this gamma secretase complex. Two parts are then separated, the C-terminal fragment that stays in the cell and the A-beta monomers here are transported out of the cell. And so these monomers continue to aggregate out of the cell and form those extracellular plaques made of A-beta and specifically A-beta 42. It leads to for some reason, it leads to the tau hyperphosphorylation, decreased blood flow, and synaptotoxicity. And in this panel B, you can see kind of the accumulation of the A-beta plaques shown in red, both laterally and medially as the disease progresses neuropathologically. So you could see that the parietal lobes are initially affected and more so in the medial aspects in an area called the precuneus. And as we continue on, it really takes over the entire parietal, occipital, kind of temporal regions, along with the frontal lobes as well. This blue is just the phosphorylated tau that continues to progress posteriorly as well. And it's really shown here on how it progresses with the disease mechanism too. So again, kind of the tip of the temporal lobe, and then goes posteriorly, continues to accumulate with a darker blue here, and then eventually it will take on this frontal lobe, which is why there's a lot of behavioral problems later on in the disease course. But this shows you kind of the molecular aspects of the phosphorylated tau, where the tau seeds, and then it changes the conformation of these native monomers and then forms paired helicals intracellularly. So within this neuron, you see the neurofibrillary tangles, and these are passed on to the neighboring neurons transsynaptically. I see this kind of as the zombie effect, you know. So one zombie affects another, and it continues to propagate, and that's what's going on with the phosphorylated tau. Now, the genesis of the plaques and tangles, it's been about 116 years to be exact since Dr. Alzheimer's actually described the neuropathologic changes. And after all this time, we actually still don't know the genesis of these plaques and tangles. We know kind of the mechanism of APP and p-tau, but we don't know the inciting event. And I think that it really dawned on me that this is the case. As a medical student, I thought I was missing something big. But only recently did I realize there are so many hypotheses out there that are trying to determine the genesis of the plaques, so the genesis of the tangles, and it really tells us that we don't have a clear grasp just yet. You know, for example, Rudy Tanzi thinks that the plaques are due to an infection. You know, and it's a reaction to this infection that causes these plaques. I think that there's a genetic cause for these tangles, you know, with TSC. That's the relationship there. You know, there's some people that think gingivitis is the inciting event for the plaques and tangles. So there's a whole host of theories out there, and hopefully we'll be able to get to the bottom of it. But we're not there quite yet. Now, clinically, though, this is the most relevant thing, and this is what we base our biomarkers on. So for the x-axis, you can see that there's no disease here, and we use something called the CDR, or Clinical Dementia Rating Scale, to determine someone's clinical or functional status. 0 means they're still normal. 0.5 means they have equate to about a MCI diagnosis. 1 is early dementia. 2 is moderate, and then 3 is advanced stages of dementia. But as we typically see patients right here or right here in this kind of pinkish light red area, you can see that the A beta deposition has significantly accumulated here in the red line. And then the phosphorylated tau, by the time they see us, has already accumulated as well, which is why the biomarkers are so helpful. You know, in someone with MCI, very mild symptoms, it's not affecting their daily function yet. Is it because of the A beta and the p-tau? And then the other 2 lines, you can see the synaptic toxicity is pretty significant as well. So this goes down pretty significantly as the clinical course continues, and then the microglia, or just the glia in general, begin to activate as the disease course continues. So keep this in mind. This is what I think of from a neuropathologic standpoint when I'm seeing patients in clinic, is that where on the spectrum do they lie in terms of neuropathology, and where on the spectrum do they lie clinically and cognitively as well. Now, what's very interesting, though, is more recent studies have shown that the incidence of Alzheimer's disease is actually decreasing. So this is actually good news. Globally, you can see the previous trends were thought to have kind of an exponential increase shown in the solid red line. But the more recent data shown in the dash red line globally shows that there's been a flattening of the curve, if you will. So especially since about 2020, it's really separated, and it continues to separate. And that's a good sign for Alzheimer's disease patients or potential Alzheimer's disease patients in general. When you go down here, when they parse it out between the global population and people from Europe and the United States, these countries are typically known more to have a higher socioeconomic status. You see just in general, the incidence is actually quite low, but it's even lower now based on the current projected numbers, which is a great thing. Unfortunately, though, the prevalence continues. So as you can see in this map, we're doing a pretty good job. We're, 12 to 17% here. Unfortunately, the neighbors need a little help that are surrounding us. The Southwest is also quite heavy in terms of the prevalence of Alzheimer's disease, and a few other spots such as Alaska and Vermont too. Now, the prevalence of underrepresented minorities, though, unfortunately has not changed. So I think that's kind of the contrast that I want to point out today, is that although the incidence is decreasing, the prevalence remains quite high for underrepresented minorities. In the African-American population, 18.6% are affected with this disorder. In the Latinx population, 14%. And to compare, people of European American descent have about a 10% rate of developing Alzheimer's disease. Now, the increase in prevalence of Alzheimer's disease is thought to be due to various other comorbid conditions that are contributing to the Alzheimer's disease pathology. So, especially in the African-American population, we know that the vascular risk factors are far more prevalent along with CKD, chronic kidney disease. And so that is thought to actually be contributing to the Alzheimer's disease pathology. Now, the socioeconomic risk factors are something that I'm going to focus on today, and I think that's something that is hard to measure biologically, and I think we actually now have a measurement for that, and I'll talk about that a little more. And then there's specific genetic differences. I think I talked to Mike Lutz about this a few times, and even the autosomal dominant mutations that we see in Alzheimer's disease for the African-American population are not the same when compared to the ones in European Americans. So the biology is actually different from the genetics and onward. So I'd like to show you this as well, the ApoE genotyping in underrepresented minorities, and specifically in the African Americans. You can see there's far fewer ApoE33s when compared to European Americans. And I think there's a disproportionate number of E4s in the subsequent categories, as you can see here, E34 and even E32. And so Dr. O'Brien has talked about that E3 is not neutral. It just means that it's a delay in the development of this disease later on. And as you can see, the E44, it's almost double the rate, but also the E22 is almost double the rate. And so it's just kind of a mixed result in terms of what the ApoE is telling us and the genetics in general in the underrepresented minority population. So the genetics does not fully explain racial disparities in the AD. That's the point that I'm trying to make right now. There's a lot of environmental cues that I think are unexplained. And I think this really points towards an epigenetic phenomenon, which I'm going to talk about now. So epigenetics, what does it mean? Epigenetics simply means modification of gene expression without altering the DNA base pairs. So there's a lot of mechanisms for it. There's the methylation, that's the most common one, a histone modification, and lastly, non-coding RNAs. Think of epigenetic mechanisms as the switches or the dimmers in your house. And the DNA base pairs as the wiring. So you never change the wiring of the house, but you can modify the lights with just the switches with on and off, the dimmers, so on and so forth. So that's kind of how I see the epigenetic mechanisms. And there's been a lot of research in terms of epigenetics and Alzheimer's disease over the past decade. So several papers are showing relationships with epigenetics, certain genes, and how they may develop into Alzheimer's disease, as you can see here. The problem with epigenetics, though, is number one, the mechanisms change with age. So my epigenetic profile as a fellow is different than it is now as a faculty member. So just with the last 4 years that I've been here, my epigenetic landscape has changed. Secondly, it varies with cell type. So what makes a blood cell is the epigenetic landscape there. What makes a neuron is the epigenetic landscape there. That's why all of these cells have the same DNA, yet they are completely different cell types. And then lastly, how they respond to the environmental cues. And I really want to focus on this, the environmental cues, and how it changes our epigenetics to really explain the racial disparities in Alzheimer's disease. And that's actually what makes epigenetic studies very difficult. So it makes it very difficult to kind of make a causal relationship because of the environmental cues. Is it the environment that changes our epigenetic landscape, or is it the other way around? So the chicken and egg thing again. Now, environmental stressors early in life, there's several of them. So intrauterine, puberty, and reproductive phases, they all contribute to the Alzheimer's neuropathology. So that is well established. Secondly, early stressors, obesity within the mother, obesity, depression, and then environmental changes also contribute to the prenatal stressors. And so we really want to focus on the prenatal stressors here, because there was a recent study in January of 2022 that showed people from Rwanda, women who were pregnant from Rwanda who underwent genocide, had a completely different epigenetic makeup compared to pregnant women who did not experience that genocide. And so there's specific genes that were turned on, specific genes that were turned off, leading to a lot of this prenatal stress and differences in the ICRs, or specifically the epigenetics. So imprinted genes is what I'd like to talk about next. And so this is a concept that will introduce a novel kind of epigenetic mechanism as well. So epigenetic imprinted control regions are regions that are methylated within the DNA, and they control imprinted genes, of course. And so imprinted genes are the ones that control your hair color, your eye color, your height, your personality. You know, you have your mother's eyes, you have your father's personality. Those are imprinted genes. And there's these specific regions called imprinted control regions, ICRs, that control these genes. There's one ICR approximately controls about 5 genes. And so keep that in mind as we go forward. But there's a lot of appealing aspects of these imprinted control regions that I'll talk about here in a little bit. Now, the ICRs specifically, there's many specific characteristics that make it very appealing to study the environmental changes. Number one, it's established prior to gastrulation. So all these stressors that your mother or your father actually endured is erased during the fertilization phase. And so all this methylation status is erased once the egg and the sperm meet. But then during this gastrulation period, this short critical period, everything is remethylated. And so that's a critical aspect that I want you to keep in mind, because that short period will help us determine what stressors were going on with the mother and the baby at that time. It's identical in all cell types. So if these are established before gastrulation or the development of all germ cell layers, so the endoderm, the mesoderm, the ectoderm, all cell types should have the same type of imprinted control regions throughout. And then lastly, they maintain the signature in all somatic cells throughout life. This makes it a potential biomarker for us to determine, is there any causal relationship between prenatal stressors and the development of Alzheimer's disease later on. Now, there are 25 previously characterized ICRs, but there are specific criteria for the ICRs that I'm going to talk about right now. Number one, they have to have consecutive CPG sites. So what that means is they have to have a cytosine phosphate guanine site, at least 5 in a row. And the second one is that the methylation level has to be about 50% plus or minus 15%. And then lastly, it has to have a methylation level that's consistent through all 3 germ layers. The brain represents the ectoderm, the kidney represents the mesoderm, and the liver represents the endoderm. And so this is the data that we've shown with my collaborators at NC State. MEG3 is a known ICR site shown here in the sea green colors. And as you can see, we looked at the methylation site in the brain, kidney, and liver. And so you can see that the methylation period is about 50% in all 3 of these layers, making it consistent throughout all germ layers. And then in the sperm, you can see the y-axis just shows you the methylation status, 0 methylation to 100% methylation here, which is why the green is about 50%. The sperm has no methylation here, And then the O sites, you can see kind of a few yellow dots here that represents 100% methylation, fulfilling the criteria for the ICRs or imprinted control regions. This leads us to think that there are a few other candidate ICRs down here shown in red in this gene called MEG3. In this panel C, again, the same kind of setup, methylation from 0 to 100 in the brain, kidney, and liver. You can see it's about 50% methylation in all three of these tissues. Sperm has no methylation. The oocyte has 100% methylation, meaning it's all coming from the mother in this case, but there's no known ICR, but there's a candidate ICR here in the PTCHD3 gene. So this is a potential new ICR that has not been described as of yet. So that's kind of what we're looking at now. This is the paper that we just published a few months back, really describing the results. And this is the website that you can actually use now to determine ICRs, which may occur, and they may be associated with various other diseases as well. Now, I want to dive in a little more into utilizing these ICRs to explain the racial inequalities of Alzheimer's disease. And so the ICRs in African Americans, it's a pretty profound difference. So we use the brain bank from the Joseph and Kathleen Bryan ADRC brain bank here, and we used two cohorts, one with Alzheimer's disease and another with normal cognition. There were 9 AD patients and 8 patients with normal cognition. The brain and blood were both investigated, but unfortunately I have to tell you that the blood was derived only from the European American population, and we did not have any blood that was available from African Americans from the brain bank, unfortunately. And then we started to investigate something called differentially methylated regions. You can think of differentially methylated regions as these methylated DNA portions that may or may not fulfill ICR criteria. So if differentially methylated regions fulfill that ICR criteria that I talked about, then it's potentially an ICR. Otherwise, it's just considered a differentially methylated region. Now, I'll just go back through the ICR criteria one more time. Consecutive CPG sites, 5 or more, methylation of 50% plus or minus 15%, and then it has to have the same methylation level in all 3 germ layers. Now, the demographics and ApoE genotyping of our study, which is about to be submitted, shows here in the AD population, the age is a little older, so the median age is about 84. And as you can see, there's 5 African Americans and 4 who are European American, and the sex difference is quite equal throughout. And there's an overrepresentation of the ApoE, as you can see here. In the normal cognition, the median age is a little younger, about 10 years compared to the AD, so 74 years is here. And you can see a split between the African American and European American races here, with an equal sex distribution here, and less ApoE4 as expected. Now, we're going to talk about the DMR stratified by race. So what we found is that the differentially methylated regions in the African Americans in brain tissue showed over 31,000. This is in contrast to the ones shown in the European Americans, so only 731. So there's these differentially methylated regions, potential ICRs in the African American cohort that show a 40-fold difference. This large signals may actually be the reason that explains a lot of the racial disparities. A lot of the African American mothers are undergoing a lot more stress than we know. And that is actually well established as well. And could this change their epigenetic landscape and lead to the higher risk of Alzheimer's disease as we age? That's something that we're actively investigating at this time. And especially because I'll go into it in the next couple of slides. But the AD-related ICRs, there's a potential of 120 ICRs at this time. When we compared the blood data to the brain data, we see about 120 that needs to be further validated with more power. And so with the 120 ICRs, we think there's about 800 potential genes. And the interesting thing is that these genes have a lot of signaling pathway commonalities. And so IGFR2, I think, is one that we're actively investigating right now, but there's so many other ones that I think are very interesting. And this is just the beginning of determining how these ICRs interplay with the genes and lead to the development of Alzheimer's disease as we go forward. Now, this leads me to the project that I'm about to propose, which is the Myriad study. So the mechanisms of epigenetics, so ICRs, so imprinted control regions, to possibly explain the racial disparities in Alzheimer's disease. Because again, ICRs are static. When we look at all these ICRs, that provides us a window in terms of what happened to the mother and the child very, very early on during conception and in utero. This could be used as a biomarker because it is static throughout life and provide a mechanism for the development of novel treatments for Alzheimer's disease. I think this is the most interesting thing, though, is the public policy changes, because we know that ICRs are developed right at fertilization and before gastrulation. It's this critical period that we can say we need to decrease the stress, or we need to do this in terms of public policy changes to maintain an ICR that is similar to ones that do not develop Alzheimer's disease. So my aims include, number one, correlating Alzheimer's disease-related ICRs to cognitive test results. And so this is going to be important, because in the clinic, what we see is that people from underrepresented minorities, they do not score the same as people who are of European American descent. We don't have a biological reason, and I don't think it's based solely on race. I think it's based a lot of it on the environmental cues, a lot of the stressors that they've had to endure both in utero and throughout life. And just as a quick reminder, puberty, reproductive age, all the stressors during this period changes your epigenetic landscape. And does that contribute as well, along with the ICRs? So we're just trying to correlate the ICR changes right now with the cognitive testing. And so my hypothesis is that people with higher ICRs found in the plasma will actually correlate with worsening cognitive test scores. And that could possibly have a biological explanation for what's going on in terms of the racial disparities for cognitive testing, number one. Number two, we want to validate that the ICRs seen within the brain are also seen in the plasma as well. And we're going to look at people who are African American and people who are European American, and we'll determine the differences as we had before. But even looking at the brain ICRs, and the ICRs in the plasma is going to be very important. My hypothesis is that people who are African American and have normal cognition will have a very similar ICR profile, both in the brain and in the plasma. And their profile is going to be very different from those of European Americans, again, pointing towards the prenatal stressors and stressors that they've had to endure throughout their entire life up to that point. And lastly, compose AD-specific plasma ICR screening chips for other underrepresented minorities. This includes the Latinx population and even the Asian population. So they're highly underrepresented in Alzheimer's disease, but my hypothesis is that the ICR screening chips, we're going to see a significant overlap in the African-American population when we compare it to other underrepresented minorities as well. So, please.
African-Americans, there's higher incidence of many other diseases.
Yeah.
Does this mechanism stand out in any of them?
So it's unfortunately, we don't have a great answer to that yet, because the 1,400 ICRs that were just found, have not been studied in the other diseases, So what we do know is that vascular disease definitely contributes to the Alzheimer's disease, which is why the other comorbid conditions are leading to a higher rate of Alzheimer's disease in African Americans. So, all right. So that is it. I'd like to recognize everyone here at Duke, as well as everyone here at NC State. And I want to definitely give a quick shout out to Carolina Quiroga. So she's a wonderful colleague, and I wish the best for her right now. I'm happy to take any questions. Thank you.
Any questions from the room?
Giovanni.
In utero stressors, like and prenatal as well, so during pregnancy and HIV and things like that, and then retrospectively kind of Alzheimer's, and now there have been more findings of these patients that have HIV or anything like that, or there's so many other stressors that can take place, but I just want to know if there's been any sort of predominance.
So the question,
can you hit S just to get out of this ESC? Stop shooting. Yeah, but repeat the question.
Okay, so the question sounds like, have there been any studies in utero that correlate prenatal stressors to Alzheimer's disease, such as HIV? Is that correct? HIV specifically. Oh, HIE. I don't think there's been a specific HIE. So yeah. And so I don't think that has been looked at specifically, but we're looking specifically at Alzheimer's disease at this time. My collaborators at NC State are looking at other stressors, such as toxic exposures to heavy metals, and so it's an ongoing study. And so I can't give you a definitive answer yet. I think that's still undiscovered. So yeah.
Take one from the internet. Andy, Mike Lutz, do you want to ask your question?
Yeah, sure. Andy, great talk. I was curious, do the ICRs combine in an additive sense? So thinking about putting together something analogous to a polygenic risk score?
Yeah. Yeah, no, absolutely, Mike. I think that's a great question. I think the ICRs are definitely contributing. Now, what is the major player? I'd like to think the ICRs, but I can't say for certain just yet. That's yet to be determined. The other epigenetic mechanisms are definitely at play as well, and there's been literature already. So how the environment changes as a child develops or during puberty, I think they're absolutely contributing as well. And I talk about this all the time in the clinic, where people who have sleep apnea that's not treated, that's definitely contributing. Now, whether they're major or minor players, I don't know. So,
any questions in here? We'll alternate.
I know you mentioned that the incidence of Alzheimer's has increased.
Yeah.
Is there research ongoing as to why that is the case? Is that your generations?
Yeah.
Effective. And I know there's another question that is popular thought that everyone aged enough, they would eventually get Alzheimer's.
Yeah, sounds good. So the first question was, what are the factors that are contributing to the decrease in the incidence of Alzheimer's disease? And a lot of it has to do with the vascular risk factors, controlling blood pressure, controlling hyperlipidemia, diabetes. I think that's really starting to play a role, okay? And that's why I'm trying to show you this table again, which is why the countries with a higher socioeconomic status have a lower incidence of Alzheimer's disease because they're better controlled, So that's one. And then the second question was, can you repeat that one again?
Yeah, just the popular thought that notion that if everyone aged long enough, that they would eventually get Alzheimer's. They live long enough to develop those plaques.
So we do know that the Alzheimer's disease pathology does accumulate. That doesn't mean you'll get the clinical syndrome. So about 20 to 30% of patients who are cognitively normal, who are 80 or above, actually have the amyloid plaques. but they don't have the clinical syndrome. So will you have the amyloid pathology? It sounds like you possibly will, depending on other comorbid conditions, but will you actually develop the clinical syndrome? I don't know of a study that has gone that far out. We look at the super agers 90 and above who don't have cognitive problems, but I wouldn't say that Alzheimer's disease is an eventual thing for everyone. You might get it at 80 and 90, you'll get other pathologies, such as TDP for frontotemporal dementias and things like that. But frank Alzheimer's disease is probably not going to be the case. So
none of us get out of this alive. All right, from the internet, Nada.
Thank you, Andy. That's a really great talk. My question is, what vascular risk factors are a definite association with Alzheimer's disease? I mean, you mentioned that with decreasing hypertension, diabetes, hyperlipidemia, that's maybe associated with a decreased risk of Alzheimer's. Are these well-established risk factors, and what would be the mechanism? Are we talking here about the interaction of vascular dementia or the actual effect of these factors on the Alzheimer's pathology?
Yeah, I wish I had a better answer for you, but yeah, we do know that the vascular risk factors are playing into the Alzheimer's disease pathology itself. The mechanism I am unaware of. So we do know that uncontrolled vascular risk factors will, of course, lead to strokes, but will also lead to an accelerated rate of Alzheimer's disease, which I've seen in the clinic as well. So
I will sharply disagree with what Andy said there. The data that vascular risk factors change the Alzheimer's pathology part, they definitely increase dementia, right? But whether they increase the amount of amyloid, there's only a single study that's ever shown that, and that was Rebecca Gottesman. And so now I'd refer you to Rebecca's study, which was in JAMA in maybe 2019. So Rebecca Gottesman. So it's a very important study, but the only one that ever said vascular risk factors change the Alzheimer's equation. But remember, vascular risk factors are also metabolic changes, right?
You're right.
So it depends on how you name
How you look at it.
Right. Question in the audience? Go right ahead.
Yeah, thank you so much for the talk, Andy. I did want to maybe get back to the first question that was asked, because it seems like you're using race as a bit of a heuristic. And I'm wondering how much data do you have about your sample pool going forward, if it is going to be continuing from the Bryant end? So, for example, are you planning to tease apart genetic ancestry from social determinants of health, environmental determinants of health, other known stressors from the particular subjects? I just don't know if you have access to those data. And if so, how do you plan to work that in?
Got it. So it sounds like the question is, because we're using African Americans as a cohort, is this based solely on race or genetics? Is that the question?
Well,
or environment.
Acknowledging that race is a social construct.
Right.
I'm wondering, are you teasing apart genetic ancestry from social histories, perhaps environmental histories as well?
Yeah. So
in that regard, there was just a study that showed in a certain American Indian population, I can't recall, ApoE is not a risk factor for dementia. So,
right.
It's complicated, right?
It is very complicated. And so, yeah, the problem of the social construct as an African Americans is very tough. I think right now, what we're trying to do is just establish these two cohorts right now, African American versus European American ancestry. That's as far as we can go, unfortunately, and then look at the ICRs at this point. And are there any differences within the group and between the groups? I think, unfortunately, that's probably the best that we can do at this point, because, yeah, it's a very sticky question in terms of this is a social construct and how do we get around this problem? Absolutely. So, Jim.
We're alternating.
Okay.
Jim, you're next after Andrew. Andrew has a question.
Okay.
Plenty of time. Andrew? You're either gone.
Yeah, thanks. Actually, my question was a lot like Len's, because I was trying to figure out, and I don't want to duplicate it, but trying to get to the question of African American as an identified social group. And we know that if you do a study like this in West Virginia, the African-American population in West Virginia has about 60% African-derived genes and 40% European. If you do it in South Carolina, it flips and you've got about 90% African-derived genes and 10% European genes. And so there's a lot of heterogeneity, which I know you know. I'm trying to figure out how do we make these genetic conclusions and things on these social groups, which I realize probably duplicates a lot of what Len was asking.
Yeah, so I think a lot of it is because there's so much heterogeneity, we have to look at kind of the ICRs and look at the social aspects, and people who are African American, unfortunately, are undergoing a lot more prenatal stressors, and that's what we have to base it on. We don't have a great answer. It's an active discussion that we have with the group as well, but unfortunately, I think that's kind of what we're going to set, what we're going to do right now, and then we'll see what comes from it. So,
Jim.
So a couple of things. One is estrulation occurs in week 3 of pregnancy. And so really we're looking at a very small window.
Yeah.
You know, and so
right.
I'm surprised at the 20,000, you know, the 40-fold difference in the methylation rate in that population. And it seems like, and knowing that the number of African-American brains in brain banks, I think at Duke, we have a couple, you know, and maybe there's 10 or 15 in the country, or some small number anyway. But you know, since you know that these imprint control regions are consistent across tissue, I think looking at other tissue, such as blood, easily accessible, large populations, is more likely to get you some clarity than looking at a small population of brains, even though it is brain-specific disease.
Gotcha. So you think our the study that we're about to publish now should be sufficient in terms of establishing that correlation. So,
well, I think it's going to be very hard to find an AD-specific panel from a handful of African Americans,
right?
As opposed to looking at, you know, if you had 1,000 or 2,000 African Americans in a population, it might be more fruitful place to look.
But as you know, Jim, that's a challenge, right?
Blood is easy.
Blood is easy. Yeah.
Blood is easier. Yeah, no, absolutely.
But for those interested, the NIH is really now behind financially and in terms of grants about funding way more research to understand these racial differences, if that's what they are. I'd like to point out Vishal's comment just as a consideration. Daniel, you have a question?
Yeah, I mean, so is residency considered a stressful time? Because I felt that I definitely lost some of my memory cells after undergoing the neurology residency. That would be a good study population, pre and post, to study the cognitive change and epigenetic profile.
For sleep deprivation, yes, I think, but everyone in residency is so high functioning, I think it's going to be hard for me to tease that out. So, but you know.
Some more than others.
I think everyone here is pretty good. So,
Daniel, you had a question? Daniel Parker?
Yeah, so I was just curious, there are these epigenetic biological age measures, that look at CPG sites that tell you how, old someone is biologically compared to their chronological age. And I was just curious if you're aware of anyone who's looked to see, if the sites picked up by those algorithms are similar to the ICRs that, are associated with AD risk, maybe as a measure of accelerated brain aging. I know Terry Moffat and their group have looked at some of that.
Yeah, so your question is looking at the epigenetic biological clock and its relationship to ICRs. I think it's a great question too, and we're actually having Deborah Rose help out with that with her R38 grant. I think she's looking at the epigenetic biological clock and trying to correlate that with the chronological age as well to determine is there a huge difference as well, and how that plays into social stressors and changes ICRs. So complicated social question, but yeah.
Any other questions in the room? I have a question, Andy. So I know nothing about epigenetics, but my understanding is epigenetic changes can be passed on. That a change, yeah. And so, but if you said the whole thing gets erased, how does that happen?
Yes, that's the interesting thing about this is that, yeah, the pregnant woman in Rwanda who's undergoing that genocide, she's got so much stress going on, it changes it. But once she becomes pregnant, all of those differentially methylated regions are erased for a period of time once the sperm and egg come together. And then it's the entire environment at that time that determines the methylation.
But in rodents, they've shown that the changes can still be passed on, even though they stress out the mom.
Yeah.
The mom has a baby. The baby has the changes. No more stress for the baby, but the baby still passes on the changes to the next generation.
Yeah, absolutely. So
how does that happen if it all gets erased?
Yeah. So, and I think that's why there's a much larger environmental component to ICR establishment versus just passing it on from mother and father. Does that make sense?
No, but the mice is now in a happy place and it still passes it on.
Right.
But the second, third generation.
I don't know how it correlates with the mouse, but I know the mouse is not a great representation of the Alzheimer's disease model right now. So I wouldn't hang my hat on the mouse data right now.
But it could happen because genetics are complicated.
Absolutely. And that's what we're trying to tease out. So absolutely.
All right. Any more questions? So anyway, it was just a great talk. It's in its early stages, but you're young, so this is going to go a long way. So, thanks to Andy for doing this.
Thank you, guys. All right. I'm going to leave the meeting.
Yeah.